Yeah, so originally we thought, stem cells, tissue repair, regenerative medicine. So initially a lot of our efforts went to building transplantable organoids, and we've shown this with our Japanese collaborators for the gut. We've shown it for liver, and very recently actually tear glands. We could grow tear glands that can cry, and you can transplant them, and they make functional tear glands. But that actually is probably a domain that's hard to work in for an academic lab, because almost immediately the next step would be a clinical trial. And these are exceedingly expensive and time consuming. And the business model of an academic lab like mine is, we have ideas, we write grants, we get shorter money, we publish, and we move on to the next discovery. And as long as we make discoveries, there will always be good people who want to work in the lab, and there will be money to think of the next idea. But we don't really have the structure in academic organizations to go from this proof of concept, mouse to mouse transplantation, to a phase 1 trial where we would transplant. Also, the challenges would be much more in the sort of safety, regulatory worlds than it would be in the worlds where we are strong in making real discoveries. So we then realized that CRISPR actually came about in 2012. We realized that combining CRISPR with organoids essentially allowed us to make any type of organoid that we wanted. We had started to look at patient-derived organoids, so rather than just growing healthy liver, and look how the liver works, you could actually derive organoids from people with hereditary liver diseases or gut diseases, and study that disease in great detail in a human version, albeit a small version of a human organ. With collaborators in Utrecht, for instance, cystic fibrosis, it turned out to be very simple to build an organoid model for cystic fibrosis directly from patients. We have about 1,500 of these patients in Holland. For half of them, we have organoids in our biobank, and these organoids predict extremely well if a patient will or will not respond to these spectacular drugs from Vertex. Very expensive, but if they work, they're fantastic. But for a large group of the patients, it was unclear. You could not predict from their DNA, from their mutations, if they would respond to the drug. So in Holland now, all these patients, about half of the patients that don't really have access to the drugs based on the fact that they have a rare mutation, we test them in the form of organoids. If the organoids work, the patient is given the drug, and so far we hear back from the clinic that this is a 100% match. When we say in vitro it works, there is a great clinical response as well. So this, I think, is also a link towards Xylys, where very similar things are being done now for cancer, where you can grow tumors, expose the tumors to many different drugs, and then find the drugs that will kill the tumor cells of an individual patient. So this is personalized medicine. Now, in addition, I think safety toxicity could be done quite easily on human liver organoids, gut organoids, kidney organoids. Currently, this is being validated. It would be great to be able to replace animal experiments in toxicology by possibly more relevant human model systems. And any other thing, infectious disease, many labs in the past 2 years have used organoids to study COVID. Maybe one example, hydroxychloroquine is a fantastic drug when you try to inhibit the production of virus in the typical cell lines that virology labs use. These are more fibroblast-like, they're easy to work with, they infect well. Chloroquine will kill that infection very well, but as we all know, chloroquine does nothing in patients. So that was a big disappointment. Turns out that organoids model the infection much better. We've shown that, but many others have shown this now. So epithelial structures, the ones that we have in organoids, allow the virus to enter through the apical site where it binds to ACE2, to the receptor of the virus. But then it doesn't go into the cell by endocytosis. It actually fuses with the cell membrane. It uses the spike protein for that. So that entry is very different from the way the virus enters in the workhorse cell lines of virology labs. And last year had these labs not only tested hydroxychloroquine or other drugs in VRE6 cells, for instance, but had it done it in primary organoids, gut, lung, they would have noticed that chloroquine does nothing when you give it to these primary epithelia. So we think that also in the context of not only toxicology, but in the entire drug development process, from the high throughput screens to the, finding your definitive candidate, organoids are probably much better disease models if you build them well, really reflecting human disease. They're probably cheaper, a lot cheaper than animals. They're not as easy as cell lines to work with, but again, I believe they are much better in predicting what will be a good compound to take into phase 1. So I think the entire drug development process can probably be replaced by organoids if you build these disease models well.